Well, good afternoon, folks, and welcome to the fourth and final webinar in the 2018 Animal Science Research Webinar Series. I'm Chris Hostetler. I'm the Director of Animal Science for the National Pork Board. As you know, this year's webinar series has been focused on improving sow lifetime productivity, and it has highlighted the need for a better understanding of why sows leave the herd, as well as the critical nature of gilt management during development. I recognize many of the names on the caller list today, participant list today, as people that have joined for all four webinars, or at least multiple webinars, and thank you for your participation. This has been extremely well received. Last week we had over 100 registered participants. This week we were at 88 when the meeting got started here a few minutes ago. So thank you all for your participation in these webinars. This series is the vision of the Animal Science Committee as a means to disseminate the results of their completed research projects that were supported by their checkoff funds. This year marks the 5th year for these webinars. Previous webinars in this series, as well as from years in the past, can be found on our Animal Science page within pork.org. Well, today's webinar will be presented by Dr. Jason Ross. He is a Lloyd L. Anderson Professor in Physiology and an Associate Professor of Reproductive Physiology and Biotechnology in the Department of Animal Science at Iowa State University. He earned his Bachelor of Science degree in Animal Science at Iowa State and his MS and PhD in Reproductive Physiology with a minor in Biochemistry from Oklahoma State University. After leaving Stillwater, Dr. Ross started his postdoctoral research at the University of Missouri and was an adjunct faculty at Lincoln University. He joined that faculty at Iowa State in 2008 and has since been involved in over 40 peer-reviewed journal articles, has been a member of the graduate committee for over 20 graduate students. His current research focuses on how pelvic organ prolapses affect sow survivability and seasonal infertility / mitigation of strategies to impact heat stress on swine. Today, the title of Dr. Ross's presentation is Drivers of Sow Pelvic Organ Prolapse. This webinar is being recorded, and within a day or so, we'll post a link of that recording of this webinar. You'll be able to find that at our Animal Science page within pork.org. So by way of housekeeping, at the end of Dr. Ross's presentation today, we'll have time for question and answers. This has been a hot topic within our industry for the last 3 years, and so I'm sure there will be lots of questions. Don't feel abashed to post those questions to me. Type those in the lower right-hand corner of your screen in the chat box, direct them directly to me, and then I will relay those questions to Jason for his response. So with that, I think I'll turn over the presentation to Dr. Ross. Jason?
All right. Yeah, thanks, Chris, and for the opportunity to share an update on some of the things we've been working on at Iowa State University and within the Iowa Pork Industry Center. And as you can all see from this first slide here, that there's a lot of names on there, and it really has been a large group effort at Iowa State, and with faculty in different disciplines, and different backgrounds, and different philosophies and perspectives on potential putative causes of pelvic organ prolapse, and that was really part of the objective, was to start pinning down what the issues or what the potential issues contributing to pelvic organ prolapse have been. And so this is just a little bit of a timeline that you all can see that it was really back in September, almost a year ago, that during Dr. Hostetler's guidance and leadership, was able to put out a request for proposals that was focused on identification of causes of pelvic organ prolapse. And so many folks in the industry had been raising the flag on this issue for quite a while, for several years prior to then, with Dr. Hostetler's guidance and leadership, was able to get some funding to start pursuing how to identify and to pursue mitigation strategies. And because that was really a major barrier to making progress in this area, was that we didn't really have a strong understanding of what hypotheses should be investigated to pursue to mitigate prolapses in the industry. And so without a good direction to pursue, that's really where this proposal was focused on. And so we submitted a proposal to essentially identify risk factors that could be pursued as putative factors or causative factors for pelvic organ prolapse. And so many of you know, especially if you're a representative of a farm that's participating in the project, Amanda Chipman, she started in December 2017 on December 18th and immediately began working with the collaborators on and the PIs on the grant to help develop and implement a survey across the industry with numerous sow farms. And also I know Ron Ketchum and Valerie Duttlinger are on the call too. They were also very instrumental in helping us identify independent producers that could be contributing farms to the study as well. So by mid-March, we had 104 farms that were contributing data to the project, and by late April, we had made farm visits to 62 of those farms and collecting data. And I'll kind of get into what kind of data we collected and where we're at on the analysis aspect. So this is really kind of the paradigm that we've used in looking at pelvic organ prolapse. And as you can see, you know, we've recognized that the problem exists and has existed. And so we're in the process now of benchmarking and accurately benchmarking the occurrence of pelvic organ prolapse as a percentage of sow deaths that happen in the industry. We're pursuing the identification of putative causes. And by the end of this project, it was our objective to really have hypotheses that we could pursue, testable hypotheses to validate causes and risk factors of pelvic organ prolapse. Subsequent to that, and hopefully in the coming projects, will be the development and testing of mitigation strategies that are effective, and then the dissemination and implementation of those strategies throughout the industry. So that's kind of our working model on dealing with the issue from a bird's eye view. This specific project really had four main objectives, and at the bottom here you can kind of see this was a hypothesis generating project. So we wanted to collect enough information and data and analysis to help guide hypotheses that we can test regarding the causation of pelvic organ prolapse in sow farms. And so to do that, we started with establishing a network of industry partners and sow farm managers. We originally targeted, or our thought was to target 60 to 80 sow farms that could contribute to the study, and as I mentioned, we were able to get 104 farms involved. We developed a herd and individual sow survey tool to use it on farm. That used the expertise of the many faculty members at Iowa State that were on the project, as well as thoughts and perspectives from many allied industry partners as well to influence what their perspectives and thoughts on potential causes of prolapse might be. During that process, Amanda's established a strong communication and network of producers, allied industry, and university faculty and staff. You know, when we present this information, we're continually seeking feedback and perspectives on the analysis and additional things that we can look at. And so at the completion of this, we're hopeful to have a large repository of data, and in some cases, we've collected a few samples from some of the sow farms as well. So examples of the data that we collected in this process included herd factors, facility factors, management factors, some animal-based measures, and then we also are pulling data, large amounts of data where it's possible as well from prior year production and mortality records. And so as you can see, I won't go through the details of everything that we collected, but it was an extremely comprehensive survey with regard to herd health and management strategies down to whether the water system is using treated water or not. And so we collected more than we were able to use, and then we utilized what we had to be able to ask specific questions of our data set, which we're kind of still in that process even now. One of the components of the project that we implemented was with the 104 sow farms was the weekly reporting of mortality and prolapses to Amanda Chipman and the Pork Industry Center. And so one of the valuable things there is with, as you all know, with using different database management systems, the cause of death for a lot of different farms is variable, right? So it's hard to compare mortality records from different systems and that are using different databases. So essentially, we collect mortality data in a consistent format. So we have very few reasons of death, 3 of which are related to prolapse, that can be added to the sheet, so that we can kind of track how mortality is changing over time, and then also how that relates to you know, specific types of mortalities, whether it be prolapse or lameness or unknowns. And so our data represents about 104 farms across 15 states that have an inventory of about 400,000 sows. And so on the on-farm visits, we visited 62 of those farms in about a 4-month window across 11 of the states. We kept that down to a minimum of 4 people. Amanda did the lion's share of that data collection. And what we tried to do was collect information specific to that facility, as well as specific to individual sows at that farm. And so our project is really using individual sow farms as our unit of measure. And so what we needed to get was management strategies for individual sow farms, but also to collect some individual sow data on those farms that we could use as a representative value for that farm to make some of our comparisons and analyses. So that part of that survey was fairly extensive, but part of that, as you can see on this sheet here, sample sheet, is we took tail length and we measured the distance from the anus to vagina, and we also developed in this perineal region scoring system that I think is becoming an important part of the project. And so that analysis of that scoring system, as those of you that have spent any time in the sow farm, and if you walk down this and evaluate sows that are laying down in late gestation, there's a huge amount of variation on the perineal score, so the anal and vulva area on a sow that's in late gestation. And so one of our, at the time, an undergraduate student, Jamie Studer, who's on the call and is now a graduate student with our research group, helped take these pictures during a summer internship and help design a scoring system that, you know, we would say that these sows that are all late gestation, that these that score a 1 would be those that are presumed to be little or no risk of prolapse. They have very little protrusion or vulva swelling in the perineal area. A score of a 2 would be presumed moderate risk of prolapse. So you can see there's some additional bulging. The perineal area is more swollen, but it doesn't necessarily look like a prolapse is imminent. And then we also have the score of a 3, where there's substantial swelling of the vulva and perineal area, and in some cases you can even see the beginning of a prolapse even initiating. So that was kind of the 3 different scoring systems that we used, and we measured that on approximately 5,000 sows during the course of the experiment and the project, of which about 3,000 or so of those were laying down. So we decided to just use the data for sows that were laying down. So this project is one of those that's just continually in progress as we're continuing to collect the weekly prolapse data, and we'd like to have a full year's worth of data collection. The project started out as a 9-month project, but many of the partners that are contributing data have agreed to continue to try to get a full year's worth of data collected, and I think that's really important, as you'll see in a little bit, that some of the farms, it appears that there's a potential seasonal effect on prolapses, and we wanted to make sure that the data set at the end of the project had the seasonal effect if it was real captured. So these are just some of the analyses we've been involved with. The K-means clustering system, I'll share with you what we're doing with that, kind of to identify sow farms based on their prolapse trends. We've done some analysis on a few biological samples, and we're continuing to collect data as well. So where we stopped on the last large analysis was utilizing calendar week 6 through 24 trends, or week 6 through 24 of the calendar. So that's where all of this analysis has happened. We're on week 33 or 4 right now with the data collection and getting that into the data set. So that will continue to become stronger as we continue to go through the add in each week's worth of data. So this is really just a breakdown. You can see we use an anonymous farm ID that Amanda has assigned to each farm. Producers and sow farm managers know which farm that they manage, but they don't know who the other farms belong to. So that enables individuals and sow farm managers and systems to track their farms relative to the rest of the industry, so they can kind of see where things are happening for everyone else and where they fit into it from an industry perspective as well. And so really what you see is anything from about 0.25% annualized mortality up to 11% annualized mortality is what we've observed across these farms in the U.S. So that was just the pelvic organ prolapse rate. If you look at this next chart here, this shows the kind of cumulative mortality, right? So the red bars represent the non-pelvic organ prolapse mortality. Yellow bars represent the pelvic organ prolapse mortalities that happen for each farm. So you can see cumulative mortality. There's a few farms up hovering around 15 to 20%, and then there's some farms that are really quite successful here during this time period with mortality sticking around the 5 to 8%. But overall, we're running around 12 to 13% mortality for the industry, at least for these farms that are on this data set. So this kind of shows the time from early February, just the mortality trends, and you can kind of see that that's consistently running around 12 to 14% across all the farms in the study. And then when you look at the pelvic organ prolapse, that's kind of as an industry average is running around 2 to 3.5%. So you can kind of see, and it's continued to decrease, I think, just anecdotally in the last couple of weeks of the data that we've looked at. So this kind of just shows the breakdown, the overall breakdown of mortality. So why are sows dying in commercial production systems right now? And what we're seeing is that in these three bars here, the blue bar, the red bar, and the green bar, those represent vaginal or uterine prolapse, rectal and anal prolapse, or the combination of both. And so you can kind of see for the average of that week 6 to 24, you know, we were running around 23% of all sow deaths were the result of prolapse. And then lameness would represent approximately 28% of sow deaths, and then unknown causes would represent about 1/3 of them as well. So that kind of gives a little bit of a breakdown of the overall contribution of pelvic organ prolapse to overall sow farm mortality. And so it's a significant component of it, about 25% to 30% of the sow deaths that are happening right now, or at least during this phase of data collection. So one of the things that we did was just taking that the average for those 18 weeks that we collected data, and if we were to put the farms into different groups and say, okay, well, the 21 farms that had the worst amount of annualized mortality are represented by the red bars. The 21 farms that had the very best mortality are represented by the yellow bars. And then the remaining 62 farms that had kind of what would be considered the average mortality rate, this is kind of their breakdown on a weekly basis. And so what you see is the farms that are consistent, that had that lowest average, throughout time or throughout the project period, we're running around 6 to 8, a couple times up to 9, 10% on mortality. And then the farms that were struggling with mortality are running around 18 to 20%. So if you look at this same thing using the same approach to kind of visualize the trends in pelvic organ prolapse, what we see is that on the farms that we would consider to be average for pelvic organ prolapse, or the farms that have the best numbers, is that they're consistently lower, typically running about 1% or a little bit less than 1% on an annualized mortality. For the best farms, the average farms are clicking around 3% or a little bit below 3%. But where we see a big impact is on the farms that there's this 15, 20% of the farms that are having a greater issue with mortality was running around 8% at the beginning of the study, and it's been down to 5 or 6% later. So that's where we seem to have more of a seasonal effect, is on the farms that have the most incidence of pelvic organ prolapse occurring. So another way that we looked at that was working with Gustavo Silva and Daniel Linares and using a k-means clustering approach. And this strategy essentially utilizes the weekly data and utilizes that data to cluster the farms into groups based on their weekly averages. So it uses an algorithm rather than kind of the method I just described, where we take the average in the top 20, middle 60, and bottom 20. And so this strategy, also breaks the farms. It happened to break them into 3 clusters, although you can see the number of farms broken into each cluster was different than how we did it arbitrarily before. So, and we only have 94 farms represented here too, because of this approach required that we couldn't have any missing data points in it as well. And so sometimes we had a few dates or a few weeks that were missing for a few of the farms. So what we saw is that these 3 different clusters, 1, 2, and 3, were all different based on their strategy. So what you see within each cluster here, for example, cluster 1, their average is very low on a pelvic organ prolapse incidence rate. So this is portrayed as POP incidence per 1,000 sows, so it's normalized to inventory. And it shows in the black lines the data for the individual sow farms in that cluster, and then the blue line is the average for those farms. So again, I think these 9-10 farms that would be in cluster 3 are the ones that have the more severe seasonal effect. So that's something that, as we share this with you all, and if you have thoughts and perspective on that, that's something that we're trying to consider is why would some farms be subjective to a seasonal influence and other farms not? And as we share some more data here as we work through this, I would just keep that in mind as that influences how you think through and rationalize what the risk factors might be. So one of the things that we looked at initially is just establishing what's the relationship between pelvic organ prolapse and overall mortality. So if you just plot that relationship and you have annualized pelvic organ prolapse here on the y-axis and annualized total mortality on the x-axis, obviously there's a strong relationship because pelvic organ prolapse contributes to over 20% of the annualized deaths. The other way of looking at that over here, though, where the relationship is much weaker, is that when you compare the non-prolapse mortality and the prolapse mortality. So there's a weak relationship between the annualized pelvic organ prolapse as well as the when plotted against the annualized non-prolapse mortality. So that suggests that the causes or relationship, I guess, initially that cause pelvic organ prolapse would be unrelated to what's causing many of the other deaths on the sow farm. And you can kind of see here in the yellow circles, those are just examples of farms that have in some cases have a very low annualized non-pelvic organ prolapse mortality, but have among the highest in the industry on pelvic organ prolapse death. And vice versa, you can have farms that have very little pelvic organ prolapse, but are high incidence of mortality overall. So when we look at one of the other things that we looked at was just this is kind of shows you the distribution of the sow farms that are on the study. We have inventories from I think as low as 500, 600 sows per farm up to a couple that are over 10,000, and we just saw a complete lack of relationship between sow farm inventory and its relationship with pelvic organ prolapse. And I think that's important to know too, right? Because that different sow farms are managed a bit differently depending on size, and to identify that there's not a relationship there, I think is important as we work through this. One of the things that we also looked was just the effect of gestational housing, and so this kind of shows just the breakdown, if anything, as a representation of the farms that are in the spread across the U.S. that are using stalls and pins. So we had 58 farms that were using stalls, another 37 farms that were using pins that were in this analysis. There appeared to be a slightly lower amount of pelvic organ prolapse in the farms that are using stalls, although I've got some notes over here. There's still some major considerations to draw before drawing any conclusions from this slide, and that this doesn't take into account stocking density per pin size. It doesn't take into account group size for those pins or at what stage of gestation they're moved into pins. And so when you start breaking that down into those different management strategies, there's you end up with very few samples for each of those scenarios, right? So there's some that have small pins, 6 or 8 bred sows per pin, and then there was some on the study that had upwards of 300 sows per dynamic pin. So some are moved in after day 50 or 60 after the second ultrasound. Some are moved into pins after the second insemination or day 2 of gestation. So this is one thing that I like to share because people were talking about pelvic organ prolapse and its association with tail length. And one of the things after Amanda and a few others took measurements on over 5,000 tail lengths is that that's not associated with pelvic organ prolapse. So over here on the y-axis, you can see the incidence rate, pelvic organ prolapse per 1,000 sows per week. And on the x-axis there, you can see the average tail length for that farm. So we would take measurements on up to 80 to 100 farms for each farm that we visited and then track that as an indicator, as a value for that farm. So, and similarly, we saw a lack of a relationship between the rectum to vulva distance as well as it relates to prolapse. So one thing that a lot of folks have talked about, and we've looked at this a couple different ways, and I feel like this is I feel pretty confident in this piece of data. Is one of the things that we did, we went into each of the 62 sow farms that we visited, and we went into the gestation room and we looked at sow cards to actually evaluate how many sows are being induced on a are being induced. And there are farms that, as you can see over here, 0% of the sows are induced, and you have farms over here on the right side that over 80% of the sows get induced. So just different management strategies on those farms based on what's recorded on the sow cards. And we established that really does not have a relationship with the pelvic organ prolapse rate on those farms during that 18-week period of time where we were tracking mortality. Similarly, we did the same thing with farrowing management strategies. So that's another thing that a lot of folks do differently, even within systems. Sow farm managers will have a little bit different strategies on that. And so what you see in the graph on the left here is that the percentage of the sows, so if we look at sow cards on that farm, the percentage of the sows that are never sleeved. So in some systems, 80 or even in an individual farm, over 80% of those sows are not sleeved during parturition. And so on the chart over here on the right, what you see is these are the sows, the percentage of the sows in farrowing that were sleeved more than 3 times or more. And so there's also a number of sow farms that have a very aggressive farrowing management strategy. Over half of the farm or over half of the sows are sleeved more than 3 times or more. And in both of those scenarios, we saw that there's a lack of a relationship with pelvic organ prolapse. So, so we didn't see a need to continue pursuing farrowing management strategies as they relate to prolapse. The thing that we did see is that as we created those perineal scores and as we scored animals on the different sow farms that we were on, we were able to identify what you can see here on the bottom of this chart is the average perineal score for that farm. So farms that had higher perineal score averages in late gestation had a significant relationship to the incidence rate of pelvic organ prolapse. And so what you can see is that you know, the R-square of about 0.16, so maybe about 16% of the variation in pelvic organ prolapse incidence could be explained by variation in this perineal region scoring system. And so that's really where we started to hone in more and think, you know, the value of the perineal scoring is an important tool in kind of monitoring a farm and monitoring sows in late gestation as their potential for prolapse. And so this kind of speaks to that a little bit here on this next slide, where we looked at on the chart here retrospectively of all the animals that we scored. You know, then we cross-reference that with the sows that show up on the mortality sheets as we're collecting that data and entering that data. And Amanda was able to see that of those sows that scored a 3 across all the farms, about 7% of those subsequently died due to prolapse in that next during that next farrowing. Or preceding the next farrowing, whereas it's around 1% the sows that score a 1 or a 2. So it's about a 700% increase in your risk to prolapse, or if a sow scores a perineal score of 3. So why is that important? I think obviously, you know, we see this relationship between the perineal score and its association with increased risk of prolapse both at a farm level and on an individual sow level, suggesting that there's something that's happening biologically that causes this phenotype, right? And so it's not really something that it can be exacerbated, I'm sure, by events that are happening during farrowing and parturition, but it's really something that's the risk is established earlier prior to entering the farrowing room. And so now our perspective on that approach, at least, is to design experience before prolapse happens, that we can either further understand the biology of what's going on with sows that are having this perineal score, or can we develop a mitigation strategy, whether it's a specific treatment, to help us reduce the risk of prolapsing on these sows that are scoring higher and have a higher perineal score. So one of the things that we did is that Zoe Kiefer, a research associate with my group, went out with Amanda on a couple of the sow farms and collected vaginal microbiota from about 88 different sows. And then we sent that to work with Dr. Stefan Smith-Essner, who's a microbiologist at Iowa State University. And he did some initial sequencing and some kind of initial analysis. And what he was able to identify is that from 2 different farms, we had sows that were perineal score of 1, 2, and 3. And the 2 take-home points there are really that there's distinct differences in the vaginal microbiome that are present between different farms. And then more specifically, on the sows that score a 1 and a 3 at both farms, there's statistically different microbiomes, vaginal microbiomes that are present in sows that score a 1 compared to those sows that score a 3. So really the next step on that is, if we decide to continue pursuing that, is to get more samples, significantly more samples, to dive a little bit deeper on the specific species and families of bacteria that are present in the vaginal microflora, and then more in depth can evaluate the relationship to pelvic organ prolapse. So that just takes a lot more numbers than what we had to do preliminarily. The other thing we looked at when we did individual animal measures was just a body condition scoring in late gestation. And what we saw is that we just used a 3-point system where 1 was considered thin, 2 was optimal, and 3 was overweight. And one of the things, I think it was counterintuitive. I think if you'd asked me before we started the study, and most people that were involved, we would have guessed that those sows that are overweight are more likely to have a uterine prolapse. And it was actually the opposite of that. We saw that the sows that much greater percentage of the sows that were low body condition score had a higher risk, almost twice the high, almost double of them ended up prolapsing, so on a percentage basis. So in sows that were
Well, it looks like we lost Jason there for just a second. Jason, I'm not sure if you got disconnected or how that happened. Let's see. Let me go ahead and take Dustin Kendall off the mute here. Dustin, can you hear me fine?
I can now, but it kicked me off about 5 minutes ago. I had to call back in.
Okay.
Hey, I'm back on.
Okay. Hey, Jason.
I don't know what happened there. My my phone is about 1.5 feet away from me, so I just said call drop. So
all right.
Sorry about that.
No problem.
So anyway, I think where I was at, hopefully when it cut off, was just describing that the higher peritoneal score and low body condition score, that upwards almost 10% of those animals ended up prolapsing. And now we're starting, as you break it out into these different groups, you start getting fewer and fewer sample size for each of those. But that's kind of consistent with what we were seeing when we had the higher numbers as well. So, and that's also related to one of the management strategies that we talked to about the different, at each of the different farms, is whether they used bump feeding in late gestation. And we did see that those farms that used a bump feeding strategy, whether it was to all animals here or just to the animals that were in lower body condition, we saw that those two, either individually or collectively, if a bump feeding strategy was utilized, that it had a positive effect on association with pelvic organ prolapse, meaning that those farms had anywhere from 0.15 to 0.2 pelvic organ prolapse per 1,000 sows per week difference. And so up here on the right, you can kind of see, each 0.1 change in that analysis is roughly 0.5% change in annualized mortality. So bump feeding appeared to be a strategy that had a relationship with incidence rate. And so, this just kind of shows that data a little bit on in another way, in comparison it to both just pop incidence here on the left, whether there was a bump feeding strategy or not, and then over here on the right, its overall effect on mortality. And so you can see that at least in the farms that we had in this analysis, there appears to be a positive effect of bump feeding and mitigating mortality, and specifically pelvic organ prolapse. So the other thing that we saw was just a farrowing feeding strategy. So we asked a lot of questions regarding how lactation feed was provided once the sows got into lactation. And so pre-farrow here on the left, intake on pelvic organ prolapse incidence essentially was it 5 pounds or more of feed or less than 5 pounds. So, you get a lot of different answers to those questions. And so we try to break it into data that we can analyze specifically. And so we use the approach of whether they had less than 5 pounds or 5 pounds or more, because some people will turn them loose on feed. And what we saw is that the higher feed intake pre-farrow, once they're in lactation, tended to be associated with, or was statistically lower in pelvic organ prolapse compared to farms that were limiting less than 5 pounds of intake prior to farrowing. So, whereas once that sow does go on to, the other questions that we'd ask is, did they use a step-up method once the sow farrowed? Were they limit fed, or were they given ad libitum feed intake immediately after farrowing? And so the ones that use a limit approach or a step-up approach tended to be a little bit lower on pelvic organ prolapse, as opposed to the feeding strategies where ad libitum intake was provided immediately after farrowing. So why is that important? Looking at body condition score and feeding strategy, it seems to be that there's something going on with, physiologically in the sows that is related to how they're eating and how they're consuming feed prior to farrowing, based on the data set of these farms in this study. And so, we want to start looking at how can we utilize that, those management strategies, number one, to look at how can we develop a mitigation strategy, but number two, how does this information, this knowledge, influence our thinking about what the root causes of pelvic organ prolapse might be? And so one of the things that we've thought about is just, as you look at those peritoneal scores of 3, it appears that you see elevated inflammation, right? So there's significant swelling in the vulva and the anal area. And so one of the things that we looked at is it's a marker of endotoxin presence in the body is the circulation of LPS binding protein. So LPS is an endotoxin. Typically, we would measure it in a heat stress experiment, and if you have a heat stress animal and you have breakdown of the tight junctions in the gut wall, you'd have an elevation of endotoxin in circulation. And one of the physiological responses of that animal is to increase the amount of LPS binding protein in circulation. And so we looked at that in sows that we collected blood from that were peritoneal scores of 1, 2, and 3, and saw that in those sows that are 3, there's a greater amount of LPS binding protein. And so that kind of fits with some of the the feed intake, I think, in conversations with colleagues, and Lance Baumgart, who's measured LPS a significant amount and has done quite a few projects looking at the energetics and the cost of eliciting an immune response. You know, not only do sows typically or pigs go off feed during an immune response, but an immune response is extremely heavily energy dependent and glucose dependent. And so, so one of the things that happens is potentially that these sows are going off feed. That's why we see lighter body condition scores are are a higher risk yet than just those with optimal or obese body conditions. So that's something that we're continuing to look at and try to investigate in a little more specific manner as we continue to move the project forward. This is another approach we looked at, was the utilization of antibiotics on pelvic organ prolapse mortality. So this approach, the analysis here on the left, what Amanda did here was just look at, okay, the weeks that, you know, and this took a long time to dig through all the data, but on the sow farms, we just compared those weeks with antibiotics in the feed compared to the weeks where antibiotics were not in the feed, right? So typically that's going to be CTC pulse was the primary utilization of the primary drug utilizer. There's also some BMD that was utilized in the study as well. But just comparing that, we saw a difference, right? During the weeks where there's antibiotics in the feed, there was across all sow farms, there was a reduction of pelvic organ prolapse. Over here, when there was a feed pulse, we looked at this just a slightly different approach. What we have here is what we classified as before the pulse represents, I think, 3 weeks of prolapse data preceding a inclusion of an antibiotic in the feed. This is the 1 to 2 weeks that the antibiotics were in the feed, and then this is the 3 weeks following when the antibiotic pulse ceased. So, and really what you can see is that there's differences before and after compared to when antibiotics are in the feed. And so that again kind of comes back to this observation that we see pelvic inflammatory, what appears to be a pelvic inflammatory response. We also see you know, the elevated abundance of LPS binding protein in circulation, and then when you add in the utilization of antibiotics, we see a reduction of pelvic organ prolapse incidence. And so that's where really we want to move to some of the next hypotheses to test is, are these, when we're including antibiotics in a study, is that influencing the peritoneal score of sows in late gestation? And so those are some of the things that we're going to have to go back and look at again. So we did collect a lot of feed on those on 58 of the 62 sow farms that we visited. We collected a feed sample, both lactation and gestation. This is just one of the comparisons, right? And this can, you know, this analysis, we include system in the analysis, but I still think that, you know, different systems have a heavy influence on the data set. But, you know, whether it's meal or pellets, there tends to be a difference or a statistical difference in pelvic organ prolapse rate and that analysis. That's pretty straightforward analysis. But then we also looked at particle size. So obviously this would only include those farms that are using meal, not the pellet diets. And really what we saw here is that at least in the particle size represented on these farms, on the day that we collected the data, did not have a relationship with prolapse that was occurring during week 6 to 24 of this year. So, and that range was, you know, from around a little over 400 microns up to over 800 microns for the gestation diet, and around 600 to 1,000 was the range that we saw typically in the lactation diet. So didn't see a huge impact there, but again, that would not include the particle size analysis for the pelvic feed, which would presumably be smaller than the left-hand side over here with the MASH. So So we did do approximate analysis on this, and I'm just throwing this up here just to kind of show you a little bit. You know, a lot of people have talked about different factors such as calcium and phosphorus, and this is just the R-square value based on the phosphorus content of the different diets in gestation and lactation here in the green bars, are circled by the green squares, and we just saw a lack of relationship in the calcium and phosphorus in the diet as it relates to prolapse that was happening on the farm during that time frame that we evaluated it. The other thing that I think this is important is that this kind of shows where we see the industry is at on different diets as well. So, you know, just anywhere from the range of the average where people are hitting on zinc relative to what the nutritional requirements are. I think it's just a good snapshot overall for the industry that for folks to take a look at. So one of the things we had a lot of the questions and interest in dietary fat and the effect that it could have on fat and lactation. And so this is just looking at the pounds of dietary fat, not the specific source at this point, but the pounds of fat per ton in the complete feed. And so that's where we just see not a remarkable relationship, but a statistically significant relationship between dietary fat during lactation and the pelvic organ prolapse rate. So a couple other things here I'll hustle through just to get it wrapped up so we can have some time for questions. But dietary sulfur was another one. We looked at that in gestation and we looked at it in lactation. That tended to be what appeared to be a relationship between the sulfur. Again, this is one feed sample at one time point plotted against the incidence rate of a couple months. So so we got to consider that when interpreting these results as well. So yeah, this is just magnesium, gestation dietary magnesium, lactation dietary potassium, where we saw a relationship, a statistical relationship. You know, and I don't think these analysis per se are anything to dramatically jump to a conclusion on. I would advise not jumping to any conclusions on any of this analysis, as it's not the final analysis that we've gone through. But I think I'm putting this up there because there's a lot of people on the phone, and I wanted to contribute to how people think about prolapses so that collectively we can have some good ideas that contribute to surface to the top as we work through this issue for the industry. And then again, this is just the plots of the phosphorus and the calcium that I showed you in that table that was up above. So not much of a relationship there in gestation with calcium and phosphorus. So the other thing that we collected, and it was kind of a late addition to the survey, was what is the water source that farms are utilizing, and what is the treatment strategy, if any, that farms are utilizing for their water. And one of the things that we saw was that there tended to be a consistent effect on folks that were using a water treatment strategy in their sow farms tended to have fewer prolapses, right? So this is using that weekly analysis of pelvic organ prolapse from week 6 to 24 and broken down here by their water source and treatment strategy. So we had 57 farms that were using untreated well water, 13 that were using rural water, and then 13 that were using treated well water or treated pond water. And of the treatments, it was primarily peroxides, and I think all of them were peroxides and chlorides that were being utilized. And so, you know, we thought that was interesting and just decided, let's break that down. Let's look at, during this period of time, the annualized pelvic organ prolapse rate of just the farms that are using water treatment versus the farms that are not using water treatment. And what we saw there is that it was a consistent difference, not just a difference in the average, but week by week, the farms that had treated were using water treatment strategies were consistently lower with respect to annualized pelvic organ prolapse rate. The other thing then we saw, just looking at the same thing with respect to mortality, we saw depending on the week, there's one week here that kind of, there's some overlap, but generally it was consistently lower, that overall mortality was lower in farms that were using a water treatment strategy. So this is just another way of looking at that data, you know, in a similar manner, just overall treated versus not treated, and then breaking it down between those that were using chlorine as a water treatment or peroxide seem to be equally effective with respect to having a lower BOP rate compared to farms that were not using a water treatment strategy. So, and then this just breaks it down by only looking at those that were using well water. So this is the effective water treatment with well water as the source, whether it was treated or not here on the bar chart on the right. So I think that's interesting. I think it kind of fits together, you know, as we try to think of the whole data set as a whole and take things into consideration, you know, the different observations that we've seen from the management strategies through water treatment, through nutritional management, and, you know, we're starting to have some identities and areas that we should look at more specifically, design hypotheses that we should test to demonstrate causality for pelvic organ prolapse rate on sow farms, and then that will enable us to work towards the development of those mitigation strategies. So, you know, just kind of summarizing here, I feel like the project as a whole, in large part to the huge number of industry partners that have been involved with this. It's just, this absolutely couldn't have been done without a lot of good people making a commitment to help submit data and provide information in the industry to really everybody to grab a hand on the rope and help pull. So that's been, if you're a farm that's or associated with a farm or a system that's been contributing data, you know, I just want to thank you personally, because it's really helped do a lot for this project, and you know, and I think it's helped us move it a long way in a short amount of time. So we appreciate that greatly on behalf of the investigators on the project, but also on behalf of the industry as a whole. You know, and at the end of the day, we saw some analyses that we're continuing to work on this project, and I just, you know, I put down here, we just want to really work to crack the code, because I think what, you know, at the end of the day, I think most people that I've talked with would agree is that it's not a single silver bullet, or we probably would have found it by now, but there's factors that happen on a sow farm that can be additive factors, and there's factors that could be multiplying factors. And so what I mean by that is that if certain items are present on a sow farm, or factors that could be risk factors are present, that those have an additive effect that can contribute to pelvic organ prolapse when multiple factors are present. And then there's also, I think, multiplying factors that that if a couple things are happening or are the case, that's going to multiply the incidence of pelvic organ prolapse. And so it's really figuring out what are those issues, whether it's a management strategy or a water source or a dietary strategy, diet management strategy. You know, what are those factors that are additive, that can contribute to pelvic organ prolapse, and which ones of those are really multiplying that effect as well. And so as we continue to work through that with hypothesis-driven projects, I think we'll be able to really drive into mitigation strategies. So this is not a representative of everybody that's contributed. This is the folks that said, yeah, you can use our symbol, you know, and let people know that we help contribute. There's a lot of other groups that help contribute to this project as well. So, you know, it's just been a pleasure. You know, we just want to thank the National Pork Board and the Animal Science Committee and Chris Hostetler for the resources to really get this off the ground back in December. And I think we've made a lot of good traction, but it wouldn't have been possible without literally hundreds of people contributing. So thanks a lot, Chris. I'm happy to answer some questions.
You bet, Jason. Thank you for a really, really good presentation. I think this effort has been, number one, I think there's been a lot of work that's been done by Amanda and you over the last 6 months to bring this information together. But I think it's also a topic of emerging interest within the industry that we've tried to address in a strategic fashion from a monetary support standpoint. and this really does give us some additional clues into what we might be looking at in the future. So with that, there was a whole bunch of questions. I think we're over 10 questions have been sent to me so far. As I suspected, this was going to be a hot topic area. So we'll ask a few of those, folks, but if there are questions that I do not ask that you want to ask, either email me or I'm going to open this up to Jason as well. Email Jason. You can find him on the Iowa State website. Email him your question, and he can answer those questions individually. But I'll try to get to as many as we can. The first question that came in was, how can we get hold of the scoring system information?
Yeah, that's a good question. I would say just fire that off to Amanda and I for the perineal scoring. I think that's what that's based on. And really, that's designed in those slides. You know, where we tried to use a description for each of those perineal scores. You know, and it is somewhat. You know, it can vary from person to person. So for this project, the 4 of us that were on sow farms, went out to the first couple sow farms together and said, let's do this together. Let's evaluate it so we're consistently tracking data. But it is, it's not a perfect system either. And we've tried to figure out or given thought to identifying maybe some less subjective ways to look at that data or to collect that information. So they haven't developed a filter for that yet.
Yeah, maybe it's something too where the pork board could support printing of the material on similar to how we've done on you know, gilt selection and feet and leg scoring and those sorts of informational pieces in the past. So we'll talk more about that offline. So the next question is, what could be done to better classify causes of mortality, since the biggest cause was the unknown category? And there was actually a follow-on question to that. It said, survey question, how many of the unknowns were the result of leaving that field blank?
Oh, yeah, that'd be well, I'll answer the second part of that question first, and that'd be a good, my answer to that is just a good question for Amanda, because she sees all the data sheets, and I don't think there was a lot of them that are leaving it blank. And I think, you know, we gave very few options for mortality, largely because we wanted to you know, focus on, you know, pelvic organ prolapse and make sure we were identifying that accurately. But there's just so many of the other deaths that fall into that unknown category are just open to a lot of interpretation. And folks are not doing, you know, postmortems on all these animals to get us a specific answer, at least in this study. So, you know, I think that's a tough one to answer, what is contributing to an unknown death. There's because it's, you know, I don't think there's any uncaused deaths that happen, right? So there is a cause for everyone, a biological underpinning of why every sow dies on a sow farm. But I just think that what we're doing is that we're not capturing enough information on that animal beforehand to know that what a likely cause of death is. So so that's a tough one, and we've thought about that internally as well. It's like, how do we, you know, what do we need to do to start getting more specific information? And how big is that bucket, and what amount of variation is occurring in that bucket?
Thank you. Number 5 here is, housing type is confounding with genetic source as well? That was a question mark.
Yep, that would be true, yeah. So that's a tough, you know, there's definitely in all this analysis, there's some confounding effects, right? So genetic source is going to be one of those. It could be system, you know, because different systems will use some pretty consistent management practices across their farms that are in the study as well. So, you know, so that's difficult to pinpoint, you know, and we've debated that internally as well too. What's the best model? Do you normalize for all of that information, or you know, I feel like we have a good representation. In this study, we were very intentional of making sure that many systems were represented as well as many different genetic sources were represented. So I do feel like the 104 sow farms that we have are fairly representative of the industry as a whole. So
yeah.
So it makes it a little more difficult to, you know, when you're discussing how, from an analysis standpoint, which one of those factors to normalize for or to use as covariates.
Yeah, it's not as clear cut, and it's real hard to tease out in that type under the type of data collection that we've done.
Right, right.
What about mycotoxins? Is it possible to quantify effects of mycotoxins on incidence rate?
Yeah, that's also a good question as well. And so we're working on getting all those samples. We took a sub-aliquot of all those, I guess, 116 feed samples from lactation and gestation, and are submitting those for analysis, for mycotoxin analysis, and then we'll be able to go back retrospectively and compare that again. So to the pelvic organ prolapse rate. So that's a good question, and we're working with an industry partner, hopefully to get that squared away, and we'll have those have that data hopefully in a few weeks.
So that brings up another question about sort of interaction between effects. So there's a question that says, there's a mix of farm level and sow level factors that are important. Are there interactions between them? For example, is the body effect of body condition the same on high and low incidence farms?
Yeah, good question. We haven't looked at that in that manner, but that is a good question. And so, yeah, it would be interesting to go back, and those farms that would be considered in the bottom 25% or whatever, does a low body condition score sow have the same risk of prolapse as a low body condition score on a high prolapsing farm? So there's that way to look at it. And then there's the alternative is there a different percentage of low body condition scores that are happening on a low and high risk farm?
Yeah, more work for Amanda to do.
yeah. There's not a shortage of ways to look at it. I'll say that.
Well, we've reached the top of the hour, so I'm going to ask one last question here, and this sort of really gets to the crux of the matter and what it is that we're after with these webinars. But based on the outcomes of this study, what should be the first thing I should do if my farm is experiencing prolapse?
Yeah, that's a good question, and I think I'm hesitant to put a flag on it right away. One of the things I would say is to email Amanda and I and let us know that you'd be willing to participate in a hypothesis testing study, because that's really our next step, is to go out and say, okay, let's look at these other things that we've identified, right? That was the overall objective of this project, was to identify what areas we need to pursue, which ones we shouldn't. And so things that we want to pursue obviously relate to the body condition score, the bump feeding as it relates to body condition score. We want to look more into the water treatment strategies. There's some transition feeding and from gestation to lactation. So there's several things along those lines that I think we would like to look at, on those farms. And so some of those things you could start by changing management, right? And what we would like to do is work with groups that do implement any changes with respect to that type of management, so that we can look at the before and after effects of prolapse, because that's really kind of our next step, is to work with subsets of farms that we can balance across genetic sources, across systems, and ask a specific question on, if we use 12 or 14 farms and implement a management strategy in 7 of them, how does that change? Can we quantify a change in those farms by altering a bump feeding strategy? And so that's really where our next step is let's test some of these hypotheses that are related to the factors that we've identified through this study. But a great question.
Yeah. Well, there's other questions. I'm sorry that I didn't get to them, but we're at the top of the hour, past the top of the hour, folks. So again, if you have those questions and you would like them answered, either email myself or Dr. Ross, and we'll try to get you a response to those. I'd like to thank you, Jason, for your excellent presentation today on the results of your project that was funded by our checkoff dollars. If you're interested in the full report of these or other checkoff-funded research projects, you can find them by clicking on the Research tab at the top of porc.org, and then you can simply search by key terms. So if you want to know about sow longevity, you can type in sow longevity and pull up all those research projects that we've done in the past. The projects contain those search terms should pop right up. So I'd also like to remind our listeners that next year we'll once again host this webinar series each Tuesday in the month of August at 12 Central. I hope you found this information presented today and in the other webinars to be useful in improving the profitability of your enterprise, and I look forward to you joining next year's webinars. Thank you very much.